Thank you, Dr. Saleda. That's the warmest introduction I ever had. Um, I'm definitely flattered. I also feel privileged to give this presentation in this wonderful platform. Thank you for the invitation and the organizer, Alicia. Um, and also thank you all for joining the seminar. So in this presentation, I will talk about the sensor development in my lab and some interesting biological, well, observations using these four sensors. The first part of this presentation will go over the technical development, biological study in my lab in the past 1 decade. Wow, it's a long time. And then the second part would be focused on a specific project which we currently recently completed. So I hope you will enjoy the presentation. The presentation is full of picture and videos, so it's definitely some eye candy. Okay. Well, I guess I don't have to introduce too much about the cell mechanobiology. We know the cells are amazing force generator, receivers, and transducers, and also processors. On the cell membrane, there are a variety of receptors that can transmit the force, and the force would regulate cellular function. One of the receptors we are interested with is the integrin. Well, the integrin regulate cell adhesion, migration, and many other functions. And in long term, the force transmitted by these receptors would also regulate a wider range of physiological processes like the blood stemming, cancer progression, immune response, and biological development. Therefore, I mean, it is very clear that force is very important for many cellular functions. Um, but force are also difficult to study because they are intangible, they are invisible, they are elusive. In the past decades, tremendous efforts have been made by researchers in the field to develop the tools allowing us to visualize, quantify, and manipulate these forces, and therefore investigating mechanobiology. So I joined this effort when I was a postdoc in Tekjip Haas lab at the UIUC. Back then we developed a tool called tension gauge tether. So the principle is actually rather straightforward because we want to tap into this force transmission between the cell and the matrix interface. So one way to do this is to immobilize those ligands on the surface through the tethers. These molecular tethers can be designed to have a tension tolerance, meaning it can be ruptured, separated under a certain critical force. We call it a tension tolerance, kind of similar to the critical force to the molecular bond. Now, this force, well, how it works is actually quite straightforward. When the cell uses the receptor to engage the ligand, they bind to each other, transmit the force. If the force is too high or beyond the critical force of the tether gets broken, and then the force would be abolished. Overall, TGT function as a tool that can knock down the molecular force transmitted by cell. So it's a tool that can knock down the force. And then we can knock down the force and then observe whether a certain cellular function can take place or not. Uh, so yeah, it's a force knockdown tool. And we found DNA actually is a pretty awesome material for this tool development because DNA has a rather mature chemistry. Um, and also the mechanical property has been well investigated. Now at a retrospective view, and the DNA, the dissociation force actually match with the integrin tension rather well. So that's uh, another perk to use a DNA to synthesize the TGT. Now with the TGT, we investigated the cell adhesion. So The cell can adhere when the tension tolerance is high enough, higher than 40 piconewton in general. Lower than that, cell would have a very poor adhesion. So from this observation, we can deduce that integrin tension would be, well, when cell adhere, the integrin tension typically would go beyond the 40 piconewton. So overall, TGT is useful, but when you use it a lot, it's kind of a boring because TGT can only tell you one single information, whether a force is required by a certain cellular function and what is the required force level. Other than that, you don't know much, you don't get much information out from TGT. Those are spatial and the temporal information, how the force generated, transmitted, and how the mechanical transduction is taking place. TGT lack the ability to review those spatial and temporal information. Therefore, I mean, overall, uh, the better tool would be to directly visualize and see the force, right, at any time. Uh, Back then, there's a few, quite a few research labs did the pioneering work to make the molecular force sensor, to make the force visible. And these molecular sensors, they are synthesized with molecular materials. For example, Saleda's group has developed the PEG-based tension sensor, and also Dr. Dunlap developed a peptide-based attention sensor, and then later the hairpin DNA. So elastic materials has been used as a force sensor, and the force sensor is labeled by fluorophore pair or fluorophore quencher pair. The central principle is that force would change the conformation of this sensor, and then also change the fluorescence property, intensity, therefore, the force signal is converted to fluorescence signal at the single molecular level. From there, you can visualize the force and also calibrate the force range. So inspired by this pioneering work um, with the TGT, after establishing my own lab, I was thinking maybe I should repurpose the TGT into a force sensor as well. So that's what we did, and also it's quite a straightforward. Pretty much we just label the TGT with the fluorophore and the quencher, right? Um, then when the receptor bind to the ligand, force transmitted to the tension sensor separated, the fluorophore will be freed from quenching. Then you would be able to see the force. The reason why I feel it might be better idea to rename this TGT into another name when we are doing the force sensing. That is because although TGT and this force sensor share basically pretty much the same structure, but they serve for a very different purpose. TGT essentially is a force modulation tool that interfere the molecular tensions. ITS, yeah, we named it as integrative tension sensor, not a very inspiring name. But the integrative means the force sensor is irreversible. Once it is activated, the fluorescent signal would remain on the surface. That can accumulate force signal with much higher signal to noise ratio. So hence the name. Now, ITS is for force imaging. And during the application, they are kind of different as well. When you are using TGT, you want to make sure the receptor doesn't have chance to non-specifically interact with the surface. So typically people do this on a pegylated glass surface. Now for ITS application, we actually want to minimize the perturbation of ITS to the cell. Because when cells bind to ITS transmitter force, it can break this, this linker, and that can cause a perturbation. So when we are do force imaging, typically we would actually supply some permanent or stable ligand on the surface, ensure the cells have a good ligand to interact with, right? Well, anyway, we we repurposed TGD into ITS, and it worked pretty well. So now the force can be imaged on the surface. You coat the glass surface with ITS and put the cells on it. By time, the force signal would develop on the surface. It's like developing a photo. Only the picture would be a force map generated by a cell. And you can also co-image the structure. The force and the structure signal are at a pretty similar signal intensity. So the force can really just routinely imaged on the surface with a well, high sensitivity. And then you can also compare the location of the force and the structure, right? Focal adhesion here, you can see the focal adhesion leading this force track. From there, you can kind of look into the relation between force and the structure. Uh, well, there's a quick comparison between hairpin-based and also the reversible shear DNA-based tension sensor tension probes. So I would say ITS has advantages, also have disadvantages. So the advantage is signal to noise ratio is good because it accumulates signal on the surface. And also the sensor itself is rather simple. You just need a two single-stranded DNA. Anneal them together, then you're good to go. The disadvantage is that ITS typically cannot do real-time for sensing. First, it may interfere cellular functions. That's why we provide a fibronectin typically on the surface to ensure the cells can adhere normally. The ITS rupture or activation would cause minimal perturbation to the cell function. And of course, ITS is not a real-time force sensor. It accumulate historical force events and then build up the final force map. Well, we do have a few tricks that can help us to do a pseudo real-time force sensing using this ITS. So I will go over this two tricks quickly. One way to do this is to, well, do a digital processing. Now in this video, you can see a migrating cell. This is a keratinocyte that can migrate at a at a very fast speed, 20 micron per minute. So it's like a 1 micron per 3 seconds. Fast migrating cells. And on the ITS surface, they typically leave two force tracks like this, indicating the integrin tension generation. Uh, all we need to do is do frame to frame subtraction. You subtract previous fluorescence frame from the current fluorescence frame. Then you would be able to retain the current fluorescence signal generated in recent frame. Recent, for example, 10 seconds, 5 seconds, right? That's a kind of a, well, pseudo real-time force imaging. But most of the time, this is enough for us to evaluate um, what is the force signal generated in recent few seconds. From the video, we can appreciate that the force is generated, tremendous force is generated at this two side wings, rear side. Now the revelation, or conclusion is that fast migrating cells actually use a brutal force to detach the rear adhesion because, keratocyte actually have two big focal adhesions on both sides. I imagine the cell probably didn't get enough time to biochemically disassemble those two focal adhesions. So they use brutal force to peel those focal adhesions off from the surface. And the force is transmitted by the cell membrane. We did many other controls experiment to verify this is the case. Right, so force actually is used to peel those adhesions off from the surface. The second trick to do this pseudo real-time force imaging is is to use a principle like when you are seeing a movie or seeing a TV, right? We know TV, the video looks lively because the image are constantly refreshed. So put an image and they erase the old image. This principle can also be applied to ITS. Um, Here is another migrating cells, and we can see a lot of single integrin tension in the lamellopodium. Um, and we perform the image. And also, meanwhile, the image during the imaging, the laser power is strong enough to do a quick photo bleaching to kill all the old force signal. In that way, we will retain the recent force signal on the surface, uh, give you a kind of a pseudo real-time force sensing. Two facts make this strategy workable. We have been using sci-fi for the force imaging because sci-fi is really bright. It gives a lot of photons at a short time, but also it can be easily photo bleached. Um, so that make the photo bleaching or erasing process rather feasible. The second interesting feature making this strategy work is actually the protection of the quencher over the dye. It turns out when the ITS before activation, the quencher is nearby, the Sci-Fi is actually protected from the photo bleaching for some certain reason we still don't understand. But this protection made sure that ITS are safe before activation. So the photo bleaching would not bleach all the dyes on the surface. Right. Now with this single molecular force imaging and then combined with molecular localization, we also try the super resolution force imaging. It also works, right? So basically, you do the integrin tension at a single molecular level integrin tension imaging, and then localize the center, reassemble them into a force map, right? So it would improve the resolution a little bit. With the ITS, we also investigate a few interesting cell adhesion units other than focal adhesion. For example, the podosomes. We know the podosomes are punctate adhesion units with a diameter about 1 to 2 microns. This podosome have an actin core surrounded by an integrin ring, so likely they transmit the force. And indeed, on the ITS surface, we can see those force signal building up around, in the individual podosomes in this small ring pattern, right? This study actually revealed another interesting conclusion. Um, See, we know focal adhesion formation. The focal adhesion formation requires integrin tension. When cells are plated on the soft substrate, or when the integrin tension are kind of inhibited by blebstatin, then the focal adhesions won't form. However, this rule does not apply to potassium. No matter how we inhibit integrin tensions in this cell, podosomes always form with a consistent actin core and adhesion ring. This is a vinculin, right? We even put the macrophage. Oh yeah, this is a macrophage. We even put the macrophage on the pegylated glass, and the glass also under the pegylation. There's a PLL to help the cell adhere through the electrostatic force. Even without the integrin ligand, the potassium can still form properly. Therefore, we conclude that a potassium formation does not require integrin tension, although potassiums can generate integrin tensions readily, but the formation is actually tension independent, right? Let's move on to another adhesion units, which is philopodia. Philopodia is a hair-like adhesion structure commonly found during the during the cell initial adhesion stage. For example, in this video, we can see the potassium forming, and then the integrin tensions can be observed generated along the shaft of this philopodia. So it's feasible to image a single molecular force in the philopodia. And then if we do that in the non-single molecular force mode, we actually also observed quite an interesting behavior. It turns out the integrin tensions transmitted by the philopodia is not continuously distributed along the shaft. There is a there's a periodic pattern for those force distribution, and the spatial period is about 1 micron. So the conclusion is that filopodial adhesive force is discretely distributed, not continuously. Uh, we we still don't know the mechanism, so we just reported this phenomenon. Maybe saving room for future study. Um, I would say one of my favorite cells is platelets. The platelets, as we know, is blood cells responsible for stopping bleeding. In the past, we looked into the force in the platelets using the dog platelets. So this is a dog platelets. The assay is actually quite fun. You purify the platelets, and then you don't have to do anything else. Just to throw the cells on the surface, ITS surface. Then the cells will come down, adhere, generate a force gradually. So you get the force map. Now, lately, after moving into the blood center, I have the privilege to obtain platelet sample weekly, human platelets. So we moved on to study the human platelets. Like one study, we were interested about the force pattern development in the adherent platelets. So we basically fixed the platelets at a different time point and then compare image the structure, protein, and the force, right? We also looked into the stiffness sensing or stiffness response of the platelet force. It shows that platelets actually the integrin tension, the force intensity is elasticity dependent on the soft surface. The force signal is significantly reduced. But the interesting part actually is the platelet adhesion spreading are not affected much by the elasticity in this range. So they looks rather normal, but the force would be substantially reduced. Um, so that's the ITS and also some application. And we truly encountered some challenge and difficulty in the past as well during this force sensor development. Now, one challenge comes from the cell membrane bound DNAs. For example, in this video, you can see we tried a cancer cell, the breast cancer cell. Back then, we tested these cancer cells on the ITS surface, hoping to see the force signal. But this is the signal we got. Um, the fluorescent signal was developing in a very weird pattern. We call a coral-like pattern. It's nothing like focal adhesions we know about, right? Um, and it turns out indeed it's not a force signal. Uh, but I mean, we did a further study and found those signal actually was caused by the DNAs bound on the cell membrane. Because even if you remove the ligand, so the RGD peptide is the ligand we used on the tension sensor. Now, even without this ligand, you can still observe this fluorescent signal, almost similar pattern. So clearly, this is not a false signal. And what the other possibility would be helicase to dissociate the DNA, which is unlikely. And we also verified it's not a helicase. So the only possibility is the DNAs that chop off this DNA, leave the dye on the surface. So so that's the major challenge we encountered. Uh, we found cancer cells and also some immune cells, macrophage, for example. They also recruit membrane-bound DNAs to the plasma membrane. And still the open question is that why the cancer cells would recruit DNAs to the cell membrane and why this membrane DNA's activity appear in this pattern. We still don't know the mechanism at this moment. Um, And we also tested macrophage. So this is a macrophage. We know proteasome would be formed, right? And this is indeed a proteasome. If we are using the double-stranded DNA as the force sensor or just a sensor, then we would see the DNA was degraded by the DNAs in this pattern. And see, back then we talked about the Polderson force study. Actually, I didn't mention the force sensor over there is actually a DNA's resistant version, and I will get into that a few slides later. Well, anyhow, we determined that proteasomes indeed recruit the DNAs into the core region. So these core region co-localize with the DNA's activity very well. We basically kind of repurpose this into a DNA's sensor. When there is a activity happening at a cell substrate interface, this sensor can report it using the positive signal, fluorescent signal, right? Well, that's another kind of observation. We found that proteasomes and the invertipodia, oh yeah, invertipodia also recruit the DNAs. And we identified that the DNAs is DNase X. And the DNase X is the only DNase which is membrane bound. So there's really no much guess. I mean, it has to be DNase X, but of course we verified that with control experiments. Right. So that's DNase in the proteasome. And since the challenge is there, cancer cell, immune cells can recruit the DNase. Now, in order to broaden the ITS or other force sensors application, we naturally want to improve the DNase resistance for this tension sensor. So the solution is to get a DNA alternative material. For example, the peptide, the PNA, the peptide nucleic acid, right? The PNA, well, it's a nucleic acid. Well, this, yeah, pretty much like a DNA, but the backbone is replaced with a peptide. They can hybridize with DNA. They can hybridize with another PNA as well, complementary ones. Um, we use a PNA DNA duplex because we found the duplex, this hybrid duplex has already got a strong resistance to the DNAs. So the reason why we still keep one DNA strand is for the easier modification over the DNA, and also DNA is cheaper. So it's enough. This is a PNA DNA duplex. If we make tension sensor using this complex uh, duplex, the force sensor would finally be able to report the force signal in the cancer cell. So this is still the breast cancer cell. And this fluorescent signal now co-localized with vinculin pretty well, suggesting these are indeed a force signal. Right, so PNA DNA. And of course, there are other solutions as well. Recently, other labs reported using the left-handed DNA, artificial DNA to make the DNA for sensor. And they also have a superb resistance to the DNAs. Well, this is actually an unexpected perk of using PNA as a force sensor. It turns out PNA can enhance the fluorescence intensity of a dye. See here, the same Cy3 dye labeled to DNA or PNA under the same imaging settings. The Cy3 appears much brighter, actually twice brighter if it is attached to PNA structure. I think this probably is due to the PIFE effect previously reported, the protein-induced fluorescence enhancement. But I didn't expect the enhancement is quite a substantial, twice brighter. So that's one advantage. Um, okay. Lately, we also made some attempts to push on the quantitative tension sensor development. because we know DNA, the unzipping or shearing, it has a tension tolerance, has a threshold. So generally, the DNA-based force sensor is a binary sensor. It can tell you a force is higher than than a certain threshold or not, but it may not be very good to kind of respond to the exact force level at a different force level, right? So we kind of want to improve the quantitative ability. So one attempt we made is to develop this ratiometric tension sensor. This tension sensor consists of two DNA domain. Both of them would be unzipped, but they have a different length. And the theoretical prediction tells us that when the force is barely above the critical force, then the force would preferentially rupture this domain, but because it is shorter. But if the force is much higher, then somehow the force would have a similar probability to either unzip this part or unzip this part. Therefore, you can imagine when the force is high, then you would have a green signal and the red signal with a similar probability. In the ensemble mode, then you would see the fluorescence intensities are comparable in the two channels. And when the force is barely just above the critical force, then you would only get a green signal. So the fluorescence ratio can serve as a marker or indicator to tells us the average molecular force level on the local region. So that's R- RTS. And then another attempt we made is simply connect two tension sensor together in series. So well, we kind of made a name, tandem tension sensor for this connection. The principle is like this. You have a two force sensing unit labeled by different dyes. When the force is between the two critical force, then it would activate, well, say the green fluorescence. But when the molecular tension is high enough, then you would have both fluorescence signals. So in this manner, you can monitor the force at a single molecule level or ensemble mode. Both mode would be possible. Um, well, I would like to point out, I mean, concurrently and independently, all the research lab, like Dr. Minshi Yu, Dr. Salida, Dr. Liu, Dr. Ha, they adopted similar strategies to create the for sensor like this. I feel, I mean, probably we all felt the need to improve the quantitative capability for the tension sensor. So, yep. Now, that's pretty much the sensor we were developing and some biological observations. Next, I would like to focus on one specific project, the phagocytosis. So we studied the phagocytosis, the force process during the phagocytosis of a surface-bound pathogen. We know phagocytosis is a fundamental process of immune defense and homeostasis. It can engulf many different type of particles. And in the textbook, a typical picture would look like this. All the particles are suspended in the solution, and the phagocytes would use a receptor to recognize the target and bind to it, forming a cup, and then eventually take this particle into the cell body for the further process. But in reality, actually many particles, they are sticking to the matrix. Like many pathogens, they actually can have the adhesion. They can bind to the host matrix tissues. They are not that easy to be detached. And the bacteria can live in the colony as well. Cell debris can adhere to the matrix. So one question is how those phagocytes overcome the surface attachment and eventually take them off and internalize them. So that's not very well understood. Now, in order to investigate this question, this topic, obviously we want to prepare a platform that can elicit the phagocytosis. So the platform we made is actually tethers the microbeads on the surface using the glutaraldehyde cross-linking. We can tune the glutaraldehyde concentration to tune the attachment strength, right? And then we put the cells on it. The cells is transfected with a live act that allows us to see the F-actin structure. I guess, yeah, this video is rather long, so take your time and observe. I think one consistent observation would be whenever a bead is detached, before the detachment, you can see the actin wave building around the particle. Sometimes the actin wave could be even at a global scale, the whole cell scale. But eventually, the actin wave would fluctuate around the beads, and then the beads would be popped up and get internalized. Now, for those very tight attachment, the cells would actually make a multiple attempt, many attempts to build up this actin wave around it uh, until they are internalized. Now, since we know actin are closely related with integrin, right? These waves is likely related to integrin tension. So we wonder whether integrins take a part in this phagocytosis or not. Now with ITS, we can after the bead's decoration on the surface, we can coat the entire surface with tension sensor ITS and then just imaging away. So indeed, we observed the integrin tensions is a signal is building up around the beads, co-localizing with actin structure, actin ring, or actin wave, right? So the signal becomes stronger for the accumulative effect. And the tension sensor here is a pNA DNA, so it can resist the DNA's activity in the macrophage. Um, And then we also confirmed this is indeed integrin tension because if you remove the ligand on the tension sensor, you won't have this force signal. We also tested on the shear ITS, meaning the tension tolerance is higher. Then we actually didn't see strong fluorescence, indicating the integrin tension in this structure is actually between 12 and 54 picnewton. Um, and also we did a confocal 3D scan to identify where the force is generated, because it's possible the force is generated all over the beads, but that's not the case. See here, the force is consistently observed in this ring pattern at the cell substrate interface. Um, the height is truly just due to the axial resolution of the confocal. Even if you passivate the beads with casing, so ITS cannot stick to the beads, or if you stick ITS to the surface, either way, the force is only in this ring pattern, not all over the beads. Another evidence to show that the force is at cell substrate interface, not at a cell particle interface, is that say if the beads are internalized, we still see plenty of a force ring remaining on the substrate. So this indicates the force is generated at the cell substrate interface surrounding the beads, um, right? Because there's an integrin tension that suggests a new adhesion structure. Um, well, basically, we observed a new integrin-mediated adhesion structure surrounding the particles, and we name it as a phagocytic adhesion ring. Well, because it's sort of related to the phagocytosis. Okay, Then naturally, one question is, what is the integrin type in this adhesion ring? We did the immunostaining. We imaged force and immunostained the integrin beta 1 and the beta 2. Beta 2 seems to be plenty in this adhesion ring, but the beta 1 is much less. In order to verify the beta 1 antibody is truly is good, have a good quality, we also tried anti beta 1 in the HeLa cells. And indeed, focal adhesions in the HeLa cells recruit a lot of beta 1 and generate the force. So the antibody is good, meaning integrin beta 1 is truly not much in the focal adhesion ring, phagocytosis adhesion ring. If we knock down the integrin beta 1, beta 2, then the force signal is much reduced. But if we knock down the integrin beta 1, then there's no much change. Overall, we found integrin beta 2 is actually rather important for the force transmission in the phagocytic adhesion rings. Right. Uh, Other common proteins recruited to the, let's say PAR. So other proteins include taline-1, vinculin, F-actin, of course, and OP3, phosphorylated FAK, and also somehow the RAB5, which is a vesicle trafficking protein. So these are some common protein, which are also found in the focal adhesions, by the way. Well, because these PRs are quite interesting, so we want to see whether these PRs are kind of ubiquitously formed in macrophages or not. So we tried human blood monocyte derived macrophage with a different size of the beads. So indeed, you can see the force signal, right? And even if you use E. coli as the particle on the surface, the PARs and the integrin tensions were consistently generated. And then the cell lines, the human THP1 cell line and the mouse raw cell line, and even the fish, macrophage is extracted from the fish scales. All of them consistently form the PARs and also generate the force around those surface immobilized particles. So this is really consistent and robust process. We also tried this on the elastic substrate, stiff and the soft. So PARs were formed, and the force somehow it does have some response to the elasticity. The force is reduced, right? Um, then we moved on to investigate the mechanism, how this integrin tension is generated in the PARs. And we compared the mechanism to the force generation in the focal adhesion, right? So here is the control, PAR and the focal adhesion. Focal adhesion is in the HeLa cells, right? Now, if you inhibit the myosin 2 Y compound, blebstatin, they can indirectly or directly inhibit the myosin 2 activity. So in the focal adhesion, this inhibition is really potent. The force is almost completely wiped out. But it does have a very minor, it have a very minor uh, effect over the integrin tension in the PARs, suggesting myosin 2 actually is not the major, protein responsible for generating force in the PARs. Another mechanism could be the actin polymerization against obstacle, right? And integrin are supporting the actin, fix the actin wave or actin structure on the surface. Therefore, the integrins likely would be under reaction force around the particle. So we inhibited the OP2/3 complex using the CK666. Up to 3 complex actually mediate actin branching. So it's one part of a process of actin polymerization. This inhibition indeed reduces the PAR force greatly, but does not affect the force in the focal adhesion because a stress fiber formation actually is independent of up to 3 complex. If you inhibit the cells using cytochalasin D, which is a truly potent and destructive inhibitor, then the actin structures is completely disrupted, and of course, no force in both cases, right? Um, Overall, these tests shows that the integrin tension generation in the PAR is actually quite different from the force generation in focal adhesions, almost opposite effect, opposite mechanism. So actin polymerization is more important for force in the PARs, not myosin 2. Another question we asked is whether the PAR formation requires integrin tension, or what is the tension dependency. Again, we know focal adhesion formation is dependent on the integrin tension. Without the integrin tension, focal adhesions cannot be formed or matured. Uh, this actually, again, the PAR formation is a little bit different. See here, we prepared the TGT surface. Um, this surface, there's no TGT, no ligand. Well, by the way, the surface is coated with a PLL PEG. PLL help the cells to adhere through the electrostatic absorption, and the PEG kind of suppress the non-specific interaction between integrin and the substrate. Right? So this surface doesn't have any ligand. Cells still can adhere, but cells did not form the PARs. Now, if we provide the unzipped TGT, the low range of tension tolerance, right, then actually the PAR can form. By the way, on unzipped TGT, focal adhesions cannot form, but PARs can form, and you can see those are actin ring. Now, if on the shear DNA, a shear TGT surface allow a high tension tolerance, then PR formed, and the actin structure seems to be brighter. Um, Overall, if you only look into the F-actin signal, then there is a dependence. Like unzipped TGT, the F-actin structure intensity is lower. A shear TGT F-actin stronger. But if you just say whenever you see F-actin ring, you judge the PAR is formed, then by that standard, the formation of the PAR actually have a very weak integrin tension dependency, at least in this range, like a 12 to 54 range, piconewton range, the dependency is weak, much weaker than the focal adhesion requirement for the integrin tension. Right. Okay, so we kind of investigated the PAR formation, the protein components, um, and the force generation mechanism. Then more important question is, are these PARs helpful for the phagocytosis? Because PAR constantly build around the particle, so it feels like it may play a role, but we need to verify, right? So what the first test we did is to verify whether the integrin ligand on the surface is required for or helpful for the phagocytosis, because we know PAR formation requires integrin ligand. So we coated the surface with a PLL, uh, fibronectin, or collagen. And on top of it, we tether those beads. The beads could either be opsonized or passivated. And then we can evaluate the phagocytosis efficiency by tracking the bead location inside of a cell using the confocal 3D scan. For example, those beads are internalized. Uh, those beads are still on the surface. And then we can quantify the efficiency for each cell. Now this is the result. So if it is an opsonized microparticle weakly tethered, oh yeah, the tethering strength can be tuned by the glutaraldehyde concentration. If it is a weakly tethered, then cells would generally have a rather high efficiency to internalize them, phagocytic efficiency, like a percentage of the bees internalized in a cell. But if you have a strong tethering, then the efficiency is reduced, especially on the PLL surface, where the phagocytic, the PLR cannot be formed, right? And then if it is a casein passivated microbeads, then on the PLL surface, the phagocytic efficiency is even lower. And then on the fibronectin surface, collagen surface, the efficiency is much better, overall suggesting the integrin ligand on the surface seems to be helpful to promote the phagocytosis efficiency for the surface-bound particles. We also tried the E. coli. E. coli immobilized on the surface through physical adsorption or glutaraldehyde. We found that E. coli is harder to immobilize, so we used a higher concentration of glutaraldehyde. You can also monitor the E. coli phagocytosis kind of outcome, then calculate the efficiency. And again, it shows fibronectin collagen truly helped to improve the phagocytic efficiency, right? Suggesting the integrin ligands helpful, and likely PAR is the result of this integrin ligand and helped the phagocytosis. Well, that's of course that's not a decisive evidence. In order to kind of obtain more evidence to support that PAR mediate this type of phagocytosis, we also co-imaged the force and the structure, simultaneously and see what's going on during the bees detachment. Like well, this bees, right? It will be detached eventually. So you see the F-actin ring and the wave building up around it, and the force signal increase around it. If you quantify the fluorescence intensity for the F-actin and the force signal, you will see F-actin ring build up around the beads. Once the beads is detached, then the F-actin ring structure is disassembled and the force stopped to accumulate, meaning the force signal pretty much is gone, um, stopped building up. Now this is a detachable beads. If the beads are truly tight, the attachment is truly tight, they cannot be detached, then you would see the cells would produce the actin ring around the beads at multiple attempts repeatedly. And during this whole process, the integrin tension signal would build around the beads and the signal gets stronger and stronger. So the cells are truly relentlessly uh, try to detach those beads from the surface. Well, that still doesn't explain the PAR directly helped for the surface detachment. It could simply be a correlation, right? So we also looked into the F-actin ring dynamics, and we found before the particle detachment, the F-actin ring actually is constricting, becomes the inner diameter becomes smaller and smaller. And when it gets to zero, that's the critical moment the beads become detached. So this actually shows a better evidence that the F-actin ring constriction caused the beads detachment. And of course, you want to argue that maybe the beads detachment create a space for the F-actin ring constriction, right? The other way around. But that is unlikely because constriction takes about 3 minutes, and the bees detachments is just a few seconds. So we constantly see this whole process before the bees detachment. We didn't see bees detachment make the actin ring shrink in the following 3 minutes. And of course, if we inhibit the actin dynamics, we found the phagocytic efficiency is reduced greatly. Let me check the time. Oh, wow, sorry. I'm gonna move a little bit faster. Just a few more slides, bear with me. Uh, well, We also knocked down the integrin tension, and the integrin tension knockdown caused phagocytic efficiency decrease. Knockdown integrin beta 2 also caused the phagocytic efficiency decrease. So combined all the evidence, we propose that phagocytic adhesion ring is new adhesion units that help the internalization of a surface-bound particle. The general process is like this. The cells would perform the actin wave scan the surface. Once the actin wave encounter a particle obstacle, then it will be, um, yeah, well, this is pretty much last slide. I will check the questions after the presentation. Well, once this obstacle is encountered, then um, the actin transmit the force to the integrin and signal the recruitment of more integrins to strengthen these adhesion units, eventually form the PARs. And upon the PARs, actin continue to polymerize to push and squeeze into the cleavage between the particle and the substrate, and then eventually lift it up for the internalization. Right. So this is we imagined the PAR mediated phagocytosis. Right. So I truly thankful and grateful for my lab members and also the funding agency. With their support, without their support, this work cannot be done. I would also be happy to answer your questions. Um, so let me check if any question is in the chatting window. All right.
Now, I have one. This is Tim Springer speaking. That was a beautiful presentation, Xufu.
Thank you so much.
I just I have millions of questions, but we only have time for one, I think. So, and I'd really love to follow up on you with you separately on this. But my major question is, what integrin are you measuring the tension with? What is your probe to measure the integrin tension? Is it an RGD based ligand?
Correct. Yes.
Okay.
At least in this presentation, I would say all ligand is the RGD peptide.
Okay, so I'm puzzled because there's no beta 2 integrin that is known to recognize RGD.
Mhm.
Well, some of them are promiscuous. I mean,
Yep,
maybe some of the promiscuous ones can. Um, uh, can you say anything more?
of course.
Have you tried to identify what specific integrin is binding here?
Yeah. Actually, we also got this question and concern from the reviewers for this paper publication. Uh, we tried them. We actually verified from different angles again and again. Uh, it turns out there's no other conclusion. Yeah, I also checked reviewed the research protocol in the past suggesting beta 2, especially alpha M beta 2, was not known to bind with RGD peptide. But somehow on our platform, um, we found plenty of a great amount of beta 2 integrin. We even did the integrin beta 2 transfection, doing that in the live cell manner, and we knocked down beta 2, immunostain beta 2. It's almost like all the signals pointed to this conclusion that integrin beta 2 actually is the major integrin type transmitting the force. So our understanding is that perhaps under the normal circumstance, because beta 2 integrin is also a phagocytic receptor that can bind to the particle through the integrin ligand if the ligand is on the particle. But in our scenario, when the integrin ligand is on the substrate, somehow this integrin type exhibit maybe a feature that was not observed in the past. Somehow it can bind to the RGD peptide in our scenario.
Hmm.
That's our current understanding.
Hmm. Right, right. Well, I'd love to talk to you about it more offline, but there are, four different beta 2 integrins,
Hmm.
and there are small molecule antagonists and specific antibodies to many of them to block ligand binding. So I think that more follow-up there is possible. Thank you.
Thank you. Yeah, that's actually a very good question because uh, alpha amyloid beta 2 is much better investigated in macrophage, in the phagocytes. So now you mentioned there are other beta 2 integrin
Oh yeah.
complex.
Yeah, there's
Okay,
alpha X beta 2 and alpha D beta 2.
I see.
Those are also likely culprits. It could be that that you have to block all three of them,
Cool.
to completely
Yeah,
block it.
sure. Thank you for the advice.
Sure.
Great. Xuefeng, yeah, that was a great talk. I have I have a series of questions actually, all written here, but maybe I'll just in the interest of time, I'll ask just one of these things.
Mhm.
You showed so much data. One of the pieces of data that I was curious about is your filipodial, the periodicity of the
Right.
filipodial forces. Now, do you have any evidence as to whether these forces would be pointing inward or outward?
Oh, hmm. Um, no. Yeah. Well, I think the force sensor in our lab does not have the ability to report the orientation or direction for the force. Maybe combined with traction force microscopy, like for example, put the sensor on the soft substrate. That may tells us some information over the force direction. But
Yeah.
kind of in my imagination, I kind of feel the philopodia seems to be retracting. So I would imagine the force probably is pointing to the cell body, but I might be wrong.
Right. I mean, I was wondering if you had evidence from the dynamics. So essentially, do you
Oh,
see the tension mount as the Philopodia extends away from the cell, or do these forces mount after the Philopodia has already formed and engaged the substrate?
oh yeah, that's actually interesting question. Maybe we can take a look on the video. It seems like when the philopodia is developing, the force will continuously follow up. So, so yeah, it's sometimes could be hard to judge how the force signal is generated and in what kind of a spatial order.
Hmm. Yeah, the body of the cell is retracting while the Philopodia
Uh,
is extending. So I can see it is confusing. Interesting.
yeah. Well, so I cannot provide a more informative answer.
Great. I also had another question regarding your podosomes. You said something that sort of surprised me a bit. You said the
Uh-huh.
podosome formation is independent of force,
Hmm.
which is a little bit contradictory with some of the literature, including some of our conclusions. Could you say more about this, that the podosome is force independent? Is that
Yeah,
complete
of
right?
course. Yes. Yeah, I mean, see, back then we are also interested by inhibiting the force and to see whether protoson can form or not. So we tried the blebstatin and other things. We see the force is wiped out, but the protoson are still formed. That was surprising to us as well. And then we also put the cells on the so-called PLL PEG surface. So the PLL, can help cells to adhere, but does not bind to integrin. And the PEG is the basic repelling polymer. On that surface, cells can adhere very well, but integrin cannot engage with any ligand. Cells cannot form focal adhesions on such surface. However, we found podosomes were still formed pretty normally. Uh, so that's a surface can absorb the cells, but does not provide the integrin ligand. So even if you don't have a integrin ligand, the podosomes can still form. Not mention the tension. And also, yeah, I saw I saw other researchers found podosomes on the like a lipid bilayer, the fluidic lipid bilayer, right? So in the fluidic layer, fluidic surface, the potassium can also form, suggesting at least the lateral integrin tension is not required for the potassium formation. Right. That's
Right.
why we kind of we are inclined to believe that the potassium formation is force independent.
Right. But in our case, I mean, we did that work on the SLBs, right? That was our work, and it was very clear. If you changed the threshold of the rupture,
Hmm.
you tune the frequency and the dynamics of the potassiums. You change their size. We also did light triggered release of integrin adhesions, and that perturbed the potassium. So
Interesting.
we interpreted that as potassiums being force sensitive.
Hmm. Yeah, well, then I don't
Have you-
understand. I mean, that I don't know. Maybe it
Yeah.
has something to do with the exact physical property of the surface, because our experiment was mainly done on the stiff glass surface, in contrast to the your fluidic bilayer.
Right.
Maybe that can somehow affect potassium formation.
Maybe.
Yeah.
Have you tried this experiment with the 12 versus the 50 piconewton TGTs?
Oh yeah, that's probably the first thing we tried before moving to the PLL PEG surface.
And it didn't
Uh,
change the actin core or the vinculin?
it didn't. If I remember, if my memory serves well, it didn't.
Interesting.
Right. Otherwise, we won't kind of have this this conclusion actually is almost the main conclusion in this publication.
Yeah. Yeah,
Yeah.
interesting. Very interesting. I see that it's 10 minutes past the hour. We're way over time.
Yeah.
Xuefeng, that was really a tremendous presentation. I really enjoyed it.
Thank you so much.
Yeah, well again we'll thank you again and thank everybody for attending, and I really look forward to an opportunity for us to meet in person again.
There will be plenty of opportunities for me. Thank you so much, and thank you, everyone. Have a wonderful afternoon.
Thanks.